All right, welcome everybody. Um, we're going to talk about human development today. We've talked a little bit about it already in lab, but this is essentially the biological process whereby we generate an entire human being from fertilization through childbirth. It involves several different, very important processes. One is mitosis, or cell division. One is growth, where tissues or cells will change in size. Another is morphogenetic movement, where cells will move from one area of the developing embryo or fetus to another. And the final, and probably the most important, is differentiation, where the cells will change how they look and act at different points in time and depending on where they're located in the developing human being. So we'll address all of these. So we'll explain the relationship between differentiation and development, and we'll specify various stages of development. The gradual modification of anatomical and physiological characteristics during the period from fertilization to maturity is human development. These changes occur over a series of 9 months, and it's amazing to see how rapidly uh, a single cell can become a new multicellular organism in this brief period of time. In a mere 9 months, all tissues, organs, and organ systems that we've studied so far in this course take shape and they begin to function. This is one of the reasons why I actually wish that this chapter were the first chapter in A&P as opposed to the last, because this gives you an idea of where all the structures we've studied come from. What begins as a single cell, a little bit larger than a period in your book, at the end of a sentence becomes an individual whose body has trillions of cells organized into a complex array of highly specialized structures. The creation of different types of cells required in this process is called differentiation. It occurs through selective changes in genetic activity. As development proceeds, some genes are turned off and others are turned on. The identities of these genes vary from one type of cell to the other, and the patterns of expression change over time. So it begins at fertilization or conception. We can divide development into periods characterized by specific anatomical changes. Embryological development makes up the events that occur during the first 2 months after fertilization, and the study of these events is embryology, and fetal development begins at the start of the 9th week and continues until birth. Embryological and fetal development are sometimes referred to as prenatal development. Postnatal development commences at birth and it continues to maturity when the aging process begins. Although all humans go through the same developmental stages, differences in their genetic makeup produce distinctive individual characteristics. The term inheritance refers to the transfer of genetically determined characteristics from generation to generation, and the study of these mechanisms is called genetics. And we've talked about genetics in lab a little bit. We've talked about the different ways that a trait can be inherited. We've talked about simple dominant recessive inheritance, where in the case of the dominant allele, you only need one copy of the allele in order to see expression of that particular characteristic, that phenotype. Whereas in the recessive version of the allele, you need two copies present in order to see the phenotype. It turns out that that's actually not the most frequent way that characteristics are inherited. It's usually a multigenic type of inheritance where you have more than one gene contributing to a specific trait. And as a result, it gets much more difficult to assign discrete probabilities to the inheritance of these types of traits, these polygenic traits. And instead, what we do is generate a normal curve where the majority of individuals born exist in the middle of the peak of the curve and the outliers are at either shoulder. Now, keep in mind that um, every gene in the body has a basic role and a basic makeup, and there isn't that much difference between one individual and another in terms of the essential information that's contained in those genes, but there are small differences that together contribute to the diversity that we see in a human population. And it's this diversity that natural selection acts on when we're talking about who amongst a particular group of individuals will survive or not survive given a certain set of environmental conditions, say encountering a pathogen or encountering a set of uh, ecological or environmental conditions that might be deleterious to your health. And this lends robustness to the species and contributes to the survivability of sexually reproducing species. The diversity means that the species will carry on as opposed to species that reproduce asexually and as a result don't exhibit nearly as much variation in the population, so that when they encounter circumstances that are extenuating for one member of the group, the entire group is in danger. So variety is the spice of life here. So we'll describe the process of fertilization and we'll explain how developmental processes are regulated. There's our buddy the egg, and there's millions of sperm around him. You can see them all clustered. That's a real photograph. So fertilization is the fusion of the male and the female gamete that generates a zygote. At that point, we've generated a cell that contains 46 chromosomes, and not only that, a novel arrangement of the genetic material on those 46 chromosomes and a novel selection of those 46 chromosomes, distinct from either of the two parents. This is why you're not a carbon copy of either your mother or your father, because A, you're a combination of genetic material from both, and B, when we generate the sperm and the egg in meiosis, we shuffle the genes through exchange of genetic material at synapsis in metaphase of meiosis 1, and as a result, no single gamete contains a chromosome that is untouched by recombination, shall we say. So once we generate those 46 chromosomes, we're ready to build a new human being. This is where life begins, make no mistake. Once we get the sperm and the egg pronucleus to fuse, we have all the information and all the prerequisites necessary for a new human life to begin. So this is where it happens. So the functional roles and the contributions of the male and female gamete are different. The sperm delivers the paternal chromosome, so it's just a DNA delivery vehicle. It has to travel pretty far. This is why it has a flagella, because the distance from the female reproductive tract vaginal opening all the way to the oviduct is quite a distance for something as small as a single spermatozoon. In contrast, the female gamete provides all the cellular organelles, inclusions, nourishment, and genetic programming necessary to support the embryo for a week after conception. The volume of this gamete is therefore much greater than that of the sperm, and that has consequences with regard to ectopic pregnancy, as we've discussed. If you have, for instance, a constriction in the oviduct, the sperm, as you can see, are much smaller than the egg, and they can swim past the obstruction and fertilize the egg lateral to the obstruction, and that can result sometimes in an ectopic pregnancy, where the embryo will develop either in the fallopian tube or could perhaps, if the egg doesn't get drawn into the oviduct, implant in the abdominal pelvic cavity. Either of these would result in an ectopic pregnancy, which could be a disaster for mother and child. At fertilization, the diameter of the secondary oocyte is more than twice the entire length of the sperm. The sperm deposited in the vagina are already movable. They have learned how to swim in the epididymis of the male reproductive tract. And as a result of secretions in the semen, this provides the energy that the sperm use, particularly from the fructose. That's a major component of the semen. This is what the sperm use for energy to swim. The mitochondria are clustered around the base of the tail and allow them to paddle. So the first step of getting the sperm ready to fertilize the egg is capacitation. The sperm can't accomplish fertilization until they've been exposed to the conditions of the female reproductive tract. The mechanism responsible for the second step of capacitation is unknown. So just by being in the female reproductive tract, what we do is we wash away layers of the acrosome and we prepare it for its contact with the corona radiata of the egg, at which point the acrosome will release its contents and the digestive enzymes will begin to chew their way towards the egg cell membrane. Fertilization typically occurs near the junction between the ampule and the isthmus of the uterine tube within a day after ovulation. By this time, a secondary oocyte has traveled only a few centimeters, but the sperm have to cover the distance between the vagina and the ampule of the uterine tube. The sperm can propel itself at speeds of only about 12.5 centimeters per hour. So in theory, it should take a sperm several hours to reach the upper portion of the uterine tube, but the actual passage time ranges from 2 hours to as little as half an hour. What helps it out? Well, contractions of the uterine musculature and the ciliary currents in the uterine tube have been suggested as a mechanism that speeds this up. Even with the transport and assistance of the available nutrients, the passage is not easy. Of the roughly 200 million sperm introduced into the vagina in typical ejaculate, only about 10,000 enter the uterine tube, and fewer than 100 reach the isthmus. So there's a lot of attrition. And we've talked about the fact that sperm are not sentient beings. They don't respond really to stimuli. They're just like a motorboat without a pilot. They just go in whatever direction they're pointed. And if they're lucky enough to make it to the egg, then that's more happenstance than anything else. In general, a male with a sperm count below 20 million spermatozoa per milliliter of ejaculate is functionally sterile because too few are going to make it to the egg as a result of attrition along the way to fertilize the oocyte. While it's true that only one sperm fertilizes an oocyte, dozens are required in order to successfully allow fertilization to occur because they help digest away those outer layers as a result of those acrosomal enzymes. The additional sperm are essential because one sperm doesn't contain enough of these to disrupt the corona radiata and get to the egg cell membrane. In addition to that, as we saw in the Miracle of Life video, they also provide propulsion for the egg to help it move down the oviduct. So let's look at the oocyte at ovulation. Ovulation occurs before the oocyte is completely mature. The secondary oocyte leaving the follicle is in metaphase of meiosis II. It's in suspended animation, waiting for this event. The cell's metabolic operations have been discontinued, and the oocyte drifts in a suspended animation, awaiting the stimulus for further development. If there's no fertilization, the oocyte will disintegrate without completing meiosis. Fertilization is complicated by the fact that when the secondary oocyte leaves the ovary, it's surrounded by this corona radiata, this outer layer of cells here, you can see. And you might think, well, what good are these cells? What are they doing for the egg? They're protecting the egg from the female's own immune system. They serve as a sort of cloak. And because the egg is genetically distinct from the female that produces it, this allows the egg to escape detection and make its way down the oviduct without being attacked. So the cells of the corona radiata protect the secondary oocyte as it passes through the ruptured follicular wall across the surface of the ovary and into the infundibulum of the fallopian tube. Although the physical process of fertilization requires that only a single sperm contact the oocyte membrane, that sperm has to penetrate that corona radiata. The acrosomal cap of each sperm has a lot of enzymes, including hyaluronidase, that break down the bonds between the cells of the corona radiata. Dozens of sperm have to release this enzyme before the connection between the follicle cells breaks down enough to allow an intact sperm to reach the oocyte. No matter how many sperm slip through the gap in the corona radiata, normally only a single sperm accomplishes fertilization and activates the oocyte, as shown here in step 1. That sperm must have an intact acrosomal cap. The first step is binding of the sperm to the sperm receptors in the zona pellucida. This step triggers the rupture of the acrosomal cap. The enzymes inside begin to digest through the zona pellucida toward the surface of the oocyte. When the sperm contacts the surface, the sperm and the oocyte membrane fuse, and this is a trigger for oocyte activation. And it's also, as we're going to see, trigger a couple of reactions that prevent something called polyspermy. And so here you can see fertilization and activation. Once the oocyte's been activated, there are changes in the metabolic activity of the egg. The trigger for activation is the contact and fusion of the membranes of the sperm and the egg themselves. What happens next is a depolarization of the oocyte membrane because of an increased permeability to sodium. So there's a massive sodium influx when this occurs. A sudden rise in calcium levels has an important effect, including exocytosis of vesicles that are just beneath the egg cell membrane. And this is called the cortical reaction. It releases enzymes that both inactivate the sperm receptors and harden the zona pellucida, so it's more difficult for sperm to drill through it. The combination prevents something called polyspermy, which would be fertilization of an egg by more than one sperm. This would create a zygote incapable of normal development because we would have massive aneuploidy. We would have something on the order of triploidy or even quadruploidy if that were to occur. If, say, 2 or even 3 sperm were to make it in at the same time. And those eggs, even if they are generated, often end up becoming spontaneous abortions. They basically abort the human developmental process early on because of the accumulation of massive mistakes, and then that egg is lost with the menstrual flow. You never even know that it's there. What happens next? Well, there's activation of enzymes. This jacks up the cell's metabolic rate. The cytoplasm has a lot of mRNAs already stored, waiting for this event to occur. What happens as soon as we do this is that these strands are activated, they are translated, and a lot of protein synthesis occurs very, very quickly, and obviously this takes a lot of energy. Most of these proteins are required for development to proceed after oocyte activation and the completion of meiosis. The nuclear material remaining within the ovum reorganizes as a female pronucleus. And so here you can see the male pronucleus and the female pronucleus. And you might ask, what are those? Those are the first and second polar bodies. Those are meiotic products. And it's another example of cell division in which the products of the cell division are not equal. You have one product that maintains most of the cytoplasm. The polar bodies are essentially waste products and they're thrown away. While these changes are underway, the nucleus of the sperm swells as it forms the male pronucleus, and the rest of the sperm break down. And what's going to happen next is they're going to get together in a process called amphimixis. And at that point, what we've got is our first cell division, our first cleavage. Notice now that we have 46 chromosomes. We are lined up. We are ready now to begin the process of growth and development. The cell is now a zygote that contains the normal complement of 46 chromosomes, and fertilization is complete. Almost immediately, the chromosomes line up along a metaphase plate and the cell prepares to divide. This is the start of the process of cleavage, which is a series of cell divisions that produce an ever-increasing number of smaller and smaller daughter cells. This happens very, very rapidly. The first cleavage division is completed roughly 30 hours post-fertilization. It yields 2 daughters, and each are half the size of the original zygote. And these cells are called blastomeres. Now, the interesting thing about this is this rapid cleavage would occur even if we didn't introduce the genetic material from the male into the egg. You can trigger this by artificially stimulating, for instance, frog eggs in early development. So the information for these early cleavage events is already resident in the cytoplasm prior to fertilization. So it's like a pre-programmed dance that the cell goes through. But obviously we need the DNA in order to read from it later on when we start to build more complex structures. So we'll list the 3 prenatal periods and describe the major events associated with each. During prenatal development, a single cell ultimately forms a 9-pound infant who in postnatal development will grow through adolescence and maturity toward old age and death. Differentiation involves changes in the genetic activity of some cells but not others. A continuous exchange of information occurs between the nucleus and the cytoplasm of the cell. Activity in the nucleus varies in response to chemical messages that arrive from the surrounding cytoplasm. In turn, ongoing nuclear activity alters conditions in the cytoplasm by directing the synthesis of specific proteins, and in this way, the nucleus can affect enzyme activity, cell structure, and membrane properties. In development, differences in the cytoplasmic composition of individual cells triggers changes in genetic activity, and these changes in turn lead to alterations in the cytoplasm, and the process continues in a sequential fashion. let's kind of wrap our heads around this for a minute, okay? Essentially, what's happening is as these cells are dividing, they are producing products that are excreted into the extracellular environment. And those products are signals for events to happen later, basically instructions. And those instructions are conveyed to subsequent cells through an interaction between these proteins and receptors on those cell membranes. And those instructions then get sent into the nucleus, and that turns on a set of genes that contain instructions dictating how those cells are going to look and act down the road. So you might ask, well, how does this all start? How do we start with cells looking and acting different when the whole thing just looks sort of like a dividing blob of cells initially? And that's where we sort of have a misapprehension. The egg itself, even prior to fertilization, has a distribution of messenger RNA and proteins that is not equal on all sides of the egg. There are different messages at the base of the egg than at the apex of the egg. And when that egg undergoes its first cleavage, you've already generated a difference inside those two cells where one egg contains a different complement of proteins and messages than the other daughter that results from that first cleavage. And so even right at that initial division, you've generated two cells that are going to kind of look and act a little bit different from each other. And that difference is reflected in the activity of the genes in the nucleus of each of the cells. And as a result, that produces different proteins that sort of leave a breadcrumb trail for everything that comes behind it. So it's invisible to our naked eye, but you can actually demonstrate this by staining the zygote prior to fertilization. You can see the differences in the distribution of these messages and these proteins inside the egg itself. And so that sort of lays the groundwork for future differences as this mystery of human development begins to unfold. Okay? Cells begin to change through time and space because what's happening is that depending on where they are and when they're there, they're going to encounter different signals around them that'll tell them to do different things. And that results then in another series of genetic events that give subsequent instructions to cells that come afterwards. Hopefully that makes sense. If you have questions about it, ask me when we see each other in class next time, and I'll go into it in a little bit more detail. The important first step occurs before fertilization, when the oocyte's in the ovary. Before this happens, before ovulation, the growing oocyte accepts amino acids, nucleotides, and glucose, as well as more complex materials such as phospholipids and message. There you go, okay, right there. You're bringing in some message. And proteins, which are the tools that allow us to read the nucleus, the information in the nucleus, and also, in some sense, direct how and where those messages are used. These come from the surrounding granulosa cells, and because not all follicle cells manufacture and deliver the same nutrients and instructions to the oocyte, the contents of the cytoplasm aren't evenly distributed. That's what we were talking about before. After fertilization, the zygote divides into ever smaller cells that differ from one another in cytoplasmic composition. And these differences alter the genetic activity of each cell and create cell lines with diverse fates. Basically, we start doing our different dance. As development proceeds, some of the cells release chemical substances including RNA molecules, polypeptides, and small proteins. And this is the breadcrumb trail that gives different instructions to cells that come after it. Okay? And it's the interplay of those chemicals with receptors on the surfaces of these cells that dictate what a cell is going to do and when it's going to do it. And you might ask, well, how in heck do we know all this? Well, we know all this because we've actually been able to work in animal models, and we've been able to take certain gene products and certain proteins and messages and change them, and we've observed changes in the development of the organism as a result of either removing these genes or overexpressing these genes or expressing these genes in the wrong place at the wrong time. So that's a lot of the evidence that goes into what we're talking about today. So the time spent in prenatal development is known as gestation, and during the first trimester, We have embryological and early fetal development. And as a result, the rudiments of all the major organ systems appear at this time. And the second trimester is dominated by the development of organs and organ systems, which is a process that nears completion by the end of the 6th month. And during this period, body shape and proportions change, and by the end of this trimester, the fetus looks pretty human. The third trimester is characterized by rapid fetal growth and deposition of adipose tissue, so we start to see fat get laid down. Early in the third trimester, most of the fetus and major organ systems are fully functional. An infant born 1 month or even 2 months prematurely has a reasonable chance of survival because we've got most of the groundwork laid down. Earlier than that, it's really a problem because we haven't properly laid down the tissues, the organ systems, and their secretions that allow us to survive outside the womb. And so preemies have a lot of physiological problems as a result of essentially coming out of the chute before we're really ready for prime time. Okay, so next we're going to talk about... Okay, now we're going to address the first trimester. We'll explain how the 3 germ layers participate in the formation of the extraembryonic membrane, and we'll discuss the importance of the placenta as an endocrine organ. And remember back when we discussed in lab the 3 germ layers as a hallmark of embryonic development, because once these cell lines are established, it's impossible to take a cell from either the ectoderm, the mesoderm, or the endoderm, which are the 3 primary germ layers, and go back in developmental time and assume a fate that belongs to one of the other 2 remaining germ layers. And the reason for this is because those instructions have been sealed off in the genetic material through the course of development up to this point. And so even though the information is still in the nucleus, it can't be accessed to be used to make messenger RNA and subsequently protein that'll help drive those developmental pathways. So at the moment of conception, the fertilized ovum is a single cell about 5/1000th of an inch in diameter and weighing 150 milligrams. It's minuscule. By the end of the first trimester, which is the 12th developmental week, the fetus is almost 3 inches long and weighs about half an ounce. So a lot's gone on in the interim. Many important and complex developmental events occur during the first trimester. Cleavage is a sequence of cell divisions that begins immediately after fertilization, and during this time the zygote becomes a preembryo, which develops into a multicellular complex called a blastocyst. Cleavage ends when the blastocyst first contacts the uterine wall. Implantation begins with the attachment of the blastocyst to the endometrium of the uterus, and it continues as the blastocyst invades the maternal tissue. Important events during implantation set the stage for the formation of vital embryonic structures. This is when we have the implantation of this ball of cells. It contains an inner cell mass and a cytotrophoblast and a syncytiotropoblast as separate structures that are still made up of embryonic tissue, but that serve a very different purpose. The syncytiotropoblast, as we're going to find out, drills into the endometrium in order to extract nutrients for the developing embryo, pass them to the inner cell mass, whereas the cytotrophoblast forms what's eventually going to be the first of the 3 embryonic membranes, and eventually is going to contribute to the amniotic membrane and the amniotic cavity. So placentation is what happens as blood vessels are formed around the periphery of the blastocyst and as the placenta develops. This is a complex organ that permits exchange between maternal embryonic circulatory systems, and it supports the fetus in the 2nd and 3rd trimesters, but it stops functioning and is ejected from the uterus just after birth. From that point on, the newborn is physically independent of the mother. Embryogenesis is the formation of a viable embryo, and this process establishes the foundation for all major organ systems. The foregoing processes are both complex and vital to the survival of the embryo. Perhaps because the events in the first trimester are so complex, It's the most dangerous period in prenatal life. Only about 40% of conceptions produce embryos that survive the first trimester. This is when we're laying the groundwork for all the other organ systems, and any mistake during this time is going to have profound downstream effects. Remember when we've discussed pleiotropy earlier in the course, which is essentially a mistake, and because of the timing of that mistake, it leads to many downstream deleterious events. Basically, from one mistake, many follow. And the first trimester is where we can make a lot of those potential mistakes. And so this is one of the reasons they warn mothers to be particularly careful during the first trimester to avoid having these kinds of errors accumulate during the foundation of what's going to be the embryo's organ systems and vital tissues. So here we can see some of the early cleavage events that occur right after fertilization. This is a series of cell divisions that occur rapidly after the fusion of the male and the female pronuclei. The first cleavage produces a preembryo consisting of two identical cells. Well, identical in the sense that they appear the same, but to be absolutely accurate, they are not exactly identical. They have different distributions of proteins and message in those two cells that divide right after that initial cleavage. So they're identical to a point. They do contain the same genetic material. The identical cells produced by this cleavage they're called blastomeres. After the first division is completed roughly 30 hours after fertilization, subsequent divisions occur at intervals of 10 to 12 hours. During initial divisions, all the blastomeres divide at the same time, and as the number increases, the timing is less predictable. After 3 days of cleavage, the preembryo is a solid ball of cells looking a little bit like a raspberry, and this stage is called the morula. And so here you can see early and late morula. And again, if you were to look at these cells, they would appear to your eye as being identical, but in point of fact, they're not. They have different molecules in their cytoplasm, and they also would probably have different receptors on their surface at this time. The morula typically reaches the uterus on day 4. Over the next 2 days, the blastomeres form a blastocyst, which is a hollow ball with an inner cavity known as a blastocele. And here you can immediately see with the formation of the inner cell mass in the blastocele that the cells have undergone different events as a result of the creation of these different areas of tissue. The inner cell mass is eventually going to be the embryo proper. The other tissue, the blastocele, the trophoblast, and everything excluding the inner cell mass, are going to be supporting tissues for the embryo. And the fact that we've gone from a solid ball of cells to a hollow ball of cells should tell you something about the difference in behavior in different parts of this dividing mass of tissue. Essentially, to create this hollow ball, what happens is that cells at the inferior margin of the morula begin to divide more rapidly than cells near the inner cell mass, and they meet medially, sort of like a skirt hanging down from the solid ball of cells. And when they meet, they form a layer of tissue, a thin layer of tissue that's going to surround the entire inner cell mass. And so this tells you that the instructions that are being produced or followed in order to generate this type of behavior are different than the instructions involved in maintaining the inner cell mass. So obviously something different has happened at this point, and it's obvious to even the casual observer. The morula typically reaches the uterus on day 4, and over the next 2 days the blastomeres form a hollow ball with the inner cavity known as the blastocele. The blastomeres are now no longer identical in size and shape. The outer layer of cells which separates the outside world from the blastocele is the trophoblast. As the word trophoblast implies, cells in this layer are responsible for providing nutrients to the developing embryo. This is how we're going to pass important components involved in anabolism to the inner cell mass. It's going to be through what we call trophoblastic nutrition. We'll talk a little bit about more what that is. The second group of cells, the inner cell mass, is clustered at one end of the blastocyst, and these cells are exposed to the blastocele, but they're insulated from contact with the outside environment by the trophoblast. In time, the inner cell mass will form the embryo. So this is going to be you, these white cells here, at some point. So what happens next? During the formation of the blastocyst, enzymes are released by the trophoblast to erode a hole through the zona pellucida, which is then shed in a process known as hatching. The blastocyst is now freely exposed to the fluid contents of the uterine cavity, and there's a lot of nutrients in this, and so it absorbs these and provides then nutrient for the inner cell mass. These glycogen-rich fluids are secreted by endometrial glands in the uterus. Over the previous few days, the pre-embryo and the early blastocyst had been absorbing fluid nutrients from the surroundings, and this process now speeds up and the blastocyst gets bigger. When fully formed, the blastocyst contacts the endometrium and it implants. Implantation begins as the surface of the blastocyst closest to the inner cell mass touches and adheres to the uterine lining at the point of contact. The trophoblast cells then divide rapidly, making the trophoblast several layers thick. So let's hop over this. The cells closest to the interior of the blastocyst remain intact, and they form a layer of cellular trophoblast or cytotrophoblast. Near the endometrial wall, the cell membranes separating the trophoblast cells disappear, and they create a layer of cytoplasm containing multiple nuclei. And this is what we call a syncytia trophoblast. Syncytia means a single cell with more than one nucleus. In this case, it forms because of the fusion of many cells, and this is where syncytia trophoblast gets its name. So this layer erodes a path through the uterine epithelium by secreting powerful enzymes, hyaluronidase being one of them. This dissolves the intercellular cement between adjacent epithelial cells as it's released by spermatozoa during the connections between the corona radiata. This is the same enzyme doing the same trick, except in this case it's being released by the developing embryo, whereas before it was released by the acrosomal heads of the sperm. At first, the erosion creates a gap in the uterine lining, but migration and division of maternal epithelial cells repair it. By day 10, the repairs are complete and the blastocyst has lost contact with the uterine cavity. So it's completely enclosed inside the uterine wall. Basically gets buried. Further development occurs within the functional zone of the endometrium. In most cases, implantation occurs in the fundus or elsewhere in the body of the uterus, but in an ectopic pregnancy, implantation occurs outside the uterus. It can be in the abdominal pelvic cavity or even in the fallopian tubes. And as we've discussed many times before, this is a disaster for both mother and child unless the developing conceptus can be rescued and reimplanted in the uterus. As implantation proceeds, the syncytia trophoblast continues to enlarge and spread to the surrounding endometrium. The erosion of uterine glands releases nutrients that are absorbed by the syncytia trophoblast and distributed by diffusion through the underlying cellular trophoblast to the inner cell mass. So basically we're chewing things up and we're sending the nutrient liquid to the inner cell mass. We don't yet have a blood supply network that's allowing an exchange of nutrients and waste between the embryo and the mother, but we are using components of the endometrial lining as nourishment. So these nutrients provide energy needed to support the early stages of embryo formation. Trophoblastic extensions go around the endometrial capillaries, and as the walls are destroyed, maternal blood begins to percolate through the trophoblastic channels, and these areas are called lacunae. Villi extend from the trophoblast into the surrounding endometrium, and they gradually increase in size and complexity until about day 21. It's like growing roots, and you can see them here in this day 9 diagram. Now, some of the other structures, this is going to be endoderm, this is going to be ectoderm, and eventually we're going to form a cavity between these two layers that's going to be populated by mesoderm. This is going to be the amniotic cavity, and this will eventually be the amniotic membrane. This is a syncytial trophoblast, and this is what's providing nutrients to the growing inner cell mass. So notice how you got more than one nuclei in a given cell. That's why they're called syncytia. Okay, as the syncytial trophoblast spreads, it begins breaking down larger endometrial veins and arteries, and blood flows through the lacunae. Now we see some dramatic changes in what's going on in the inner cell mass. We begin to see the formation of the 3 germ layers. So formation of the amniotic cavity marks the beginning of a fluid-filled pocket that's going to protect the embryo while it develops. The inner cell mass has little apparent organization early in the blastocyst stage, but by the time it implants, it's separated from the trophoblast, and this gradually increases and creates the amniotic chamber. The trophoblast will later be separated from the amniotic cavity by layers of cells that originate at the inner cell mass and line the amniotic cavity, and these layers are going to form the amniotic membrane. When the amniotic cavity first appears, the cells of the inner cell mass are organized into an oval sheet that's about 2 layers thick. The superficial layer faces the amniotic cavity, and the deeper layer is exposed to the fluid contents of the blastocele. And so what they've done here is they've given you 2 views of the same thing, one at an earlier stage here and one at a later stage. This is day 10, this is day 12. And what you're looking at is a coronal frontal section through the developing embryo. You can see the amniotic cavity, and this is the superficial layer that's going to be eventually ectoderm, and this is going to be endoderm. This is the yolk sac, and this is going to be the amniotic membrane. So this area, believe it or not, is going to be you. Okay, these cells right in here. When the amniotic cavity first appears, it's closest to what's going to be eventually the endoderm. And we talked about the superficial layer and the deeper layer. The deeper layer here, this is the endoderm, and this is the yolk sac. And what they're showing you here is the formation of the primitive streak and the migration of cells through it, and these cells will eventually form the mesoderm. This is the space in between the endoderm and the ectoderm. And you might ask, well, how does this happen? How do these cells know to move and know where to move? And again, those are chemical signals that are left by cells that have gone before. They send signals for morphogenetic movement so that this migration can take place. So it's an example of a primitive stimulus response called positive chemotaxis, where the cell moves towards a molecule that's concentrated in the extracellular material. So next we have gastrulation and germ layer formation at day 12. So this is the formation of the mesoderm, as you can see here, as the cells migrate through the primitive streak and into this cavity. And so you might ask, well, orient me. Tell me what's going to be what. Well, at this point, what we can say is that this is eventually going to be your spinal cord and your vertebral column. So this is your dorsal side. Your ventral side is going to be here. And it's real hard to pick out anything else at this point, but we'll see as we develop more familiar structures begin to arise. So at the primitive streak, the migrating cells leave the surface. They move between the 2 existing layers, and this creates 3 distinct embryonic layers: the ectoderm, the endoderm, and the mesoderm. The ectoderm are superficial cells that didn't migrate to the into the inner cell mass. The endoderm are the layers that face the blastocele, and the mesoderm are poorly organized layers of cells that migrate through the primitive streak and populate this hollow portion of what is this disc. So these are the 3 germ layers. This is a major landmark in embryonic development. Gastrulation produces an oval 3-layered sheet, and this is the embryonic disc, and this will form the body of the embryo, whereas the rest of the blastocyst will be involved in forming the extra embryonic membranes. And so the interesting thing about these early stages is that it's possible to genetically test tissue that's going to make up the embryo at this stage through a technique called CVS, or chorionic villus sampling. So what they do is they take small bits of tissue that make up the syncytia trophoblast or the cytotrophoblast, and they can do a karyotype on it so that they can get a chromosome count, and they can also do PCR to look for defective genes, and that way they can prepare the parents for any genetic defects that might arise as a result of an aneuploidy or a known deleterious gene that they suspected may have been transmitted to their children, and they can find this out in utero, and this allows them time to prepare for any eventuality that that disease state might create after birth, postnatally. And so this is just a chart showing you the different components that come from the ectoderm, mesoderm, and endoderm, and we've discussed these already before. Ectoderm basically gives rise to the skin and the nervous system. The mesoderm primarily the musculoskeletal system, and the endoderm primarily the viscera. Now that's a sort of an oversimplification, but this chart gives you sort of a detailed look at what is composed of what. Now the interesting thing about this is that postnatally then, if you see a defect in the derivatives of any of these germ layers, you can make a fairly safe assumption that the error was made during the formation of the three germ layers, and this is why the defect would perhaps involve more than one apparently disparate set of structures, but in fact they're all related because they all come from the same germ line. So for instance, if you were to see a genetic defect that you found primarily in the skin and the nervous system, you could make a relatively safe assumption that there is a problem with the ectodermal derivatives during the developmental process. Same with musculoskeletal or the viscera. This is one of the ways that we diagnose these types of developmental disorders. Okay. Next we have formation of the extraembryonic membranes. There's three of them. Well, four of them actually. So the germ layers participate in the formation of these membranes. The yolk sac is made up primarily of mesoderm and endoderm, the amnion, ectoderm, and mesoderm, and the allantois, endoderm and mesoderm, and finally the chorion, mesoderm, and trophoblast. Although these membranes support embryological and fetal development, few traces of their existence remain in the adult system. You do find some things. They primarily exist as ligaments, but they're not major components of adult physiology. The yolk sac begins as a layer of cells spread out around the outer edges of the blastocele to form a complete pouch, already visible 10 days after fertilization. As gastrulation proceeds, mesodermal cells migrate around the pouch and complete the formation of the yolk sac. Blood vessels appear within the mesoderm, and the yolk sac becomes an important site of blood cell formation. The amnion is made up of ectodermal cells that spread over the inner surface of the amniotic cavity, and mesoderm follows that and creates a second outer layer. This is at 14 days. This combination of germ cells is the amnion. As development proceeds, the amnion and the amniotic cavity get larger, and the amniotic cavity contains amniotic fluid which surrounds and cushions the developing embryo fetus. And in point of fact, it also contains cells that are sloughed off from the developing embryo, which can also be karyotyped or used in PCR to look for defective genes or improper chromosome counts, and again, give the parents of that particular baby a chance to prepare for any eventuality that might come from these types of genetic defects. The third extraembryonic membrane, the allantois, is an outpocketing of the endoderm near the base of the yolk sac. The free endodermal tip then grows towards the wall of the blastocyst and is surrounded by a massive mesoderm. The sac of endoderm and mesoderm is found at the base of this structure. You can see it right here, the allantois. And it gives rise to the urinary bladder. The chorion is the outermost embryonic membrane. It's a tough membrane. It's associated with the allantois, and it spreads around the blastocyst, and it separates the trophoblast from the blastocele. This combination of mesoderm or trophoblast is the chorion. Find this between day 14 and 21. When implantation first occurs, the nutrients absorbed by the trophoblast easily reach the inner cell mass by diffusion, but then as things get more complex, diffusion is not going to get the job done. So as the embryo and the trophoblast get bigger, the distance between them increases. So we have to come up with another way of nourishing the embryo. So blood vessels now begin to develop within the mesoderm of the chorion. This creates a rapid transit system for nutrients, and it links the embryo with the trophoblast. The appearance of blood vessels in the chorion is the first step in the creation of a functional placenta. By the third week of development, the mesoderm extends along the core of each trophoblastic villus, and it forms chorionic villi in contact with maternal tissue. This happens between day 21 through day 70. These villi continue to get larger and branch, and this creates an intricate network within the endometrium. And this is going to be how we exchange nutrients and waste products between the maternal and the embryonic circulation. Embryonic blood vessels develop within each villus, and the blood flows through those chorionic vessels. This begins early in the third week of development when the extraembryonic heart begins beating. The blood supply to the chorionic villi arises from the allantoic arteries and veins. And I think I actually told you that your heart starts beating before it even actually has blood to pump through it. It's one of the curiosities in the formation of the circulatory system. As these villi get bigger, more maternal blood vessels are eroded. The maternal blood now moves slowly through the complex lacunae, and these are lined with syncytial trophoblast. The chorionic blood vessels pass close by, and gases and nutrients diffuse between embryonic and maternal circulation across the layers of the trophoblast. So it's very important to remember that the blood of the mother and the baby don't mix, but they come into very close contact, and they're able to exchange by diffusion nutrients and waste products. It's a size exclusion type of filtration, and as a result, anything below a certain diameter is going to be able to pass through the placenta and get into the developing embryonic tissues or fetal tissues, as the case may be. And again, this is one of the reasons why mothers are warned against exposing themselves to viral pathogens or toxins, particularly during the first trimester. So at first, the entire blastocyst is surrounded by chorionic villi. The chorion continues to get larger and expands like a balloon within the endometrium, and by the fourth week, the embryo, the amnion, and the yolk sac are suspended in a fluid-filled chamber. The body stalk, which is a connection between the embryo and the chorion, contains the distal portions of the allantois and blood vessels that carry blood to and from the placenta. The narrow connection between the endoderm of the embryo and the yolk sac is called the yolk stalk. The placenta doesn't continue to enlarge forever. Regional differences in placental organization begin to develop as the expansion of the placenta creates a prominent bulge in the endometrium. The thin portion of the endometrium, called the decidua capsularis, no longer participates in nutrient exchange, and the chorionic villi in that region disappear because you don't need them anymore. Placental function is now concentrated in a disc-shaped area, the deepest portion of the endometrium, in an area known as the decidua bassalis. The rest of the uterine endometrium, which has no contact with the chorion, is called the decidua parietalis. At the end of the first trimester, the fetus moves further from the placenta. This happens between weeks 5 and 10. The fetus and placenta remain connected by the umbilical cord or stalk, which contains the allantoic and placental blood vessels and the yolk stalk. Blood flows to the placenta through paired umbilical arteries and returns in a single umbilical vein. The chorionic villi provide surface area for active and passive exchanges of nutrients and waste products between the fetal and maternal blood circulation. And this is, in the same sense, the organization that you see in the pulmonary circulation, where the veins carry deoxygenated blood while, I'm sorry, where the veins carry oxygenated blood while the arteries carry deoxygenated blood. Remember that what we're talking about is um, blood moving to and from the embryo. So the veins are essentially um, draining oxygenated blood from the placenta and bringing it back to the embryo, while the arteries are bringing deoxygenated blood to the placenta so that it can exchange CO2 for oxygen and waste products for nutrients. Placental functions are now concentrated in the deepest portion of the endometrium, and that's the decidual basalis. The rest of the uterine endometrium is called the decidua parietal. We've already discussed this. Let's see. Okay. The blood in the umbilical arteries is deoxygenated and contains waste products generated by tissues and at the placenta. Oxygen supplies are replenished, organic nutrients are added, and carbon dioxide and other organic waste products are removed. The placenta places a considerable demand on the maternal and cardiovascular systems, and blood flow to the uterus and placenta is extensive. The endocrine placenta, in addition to its role in the nutrition of the fetus, also has an endocrine function. Several hormones, including human chorionic gonadotropin, human placental lactogen, and placental prolactin and relaxin, along with progesterone and estrogens, are synthesized by the syncytial trophoblasts and released into the maternal blood. And this, among other things, keeps the corpus luteum viable, which in turn secretes hormones that keep the endometrial lining in place in order to maintain the pregnancy. So, very important interplay. Every pregnant female has two additional endocrine organs that weren't present prior to conception, and those are the chorion, which is part of the developing embryo, and the corpus luteum, which is resident in the ovary. And they talk to each other through the blood using hormones as a communication method. Okay, we will pick this up next class, and I'll talk to you soon.